The overall goal of this procedure is to provide a sensitive technique for measuring cellular mitophagy activity using flow cytometry. Here, we demonstrate that this method was successfully used to assess the dramatic increase in HeLa cell mitophagy after treatment with CCCP, a mitochondrial uncoupler.
This technique allows mitophage activity to be measured very quickly and easily. The main advantage of this technique is that it can sensitively measure mitophage activity in living cells without any additional staining. Therefore, this technique will ultimately be of help in understanding the mechanism and physiological role of mitophage. Demonstrating the procedure will be Jee-Hyun and Yeon Kim from my laboratory.
Begin this procedure by preparing the mt-Keima, a mitochondria-targeting Keima lentivirus. First, seed HEC293T cells in a poly and lysine-coated culture dish, and culture them at 37 degrees Celsius in a tissue culture incubator for 1 day. On the next day, transfect HEC293T cells with mt-Keima lentiviral DNA and packaging DNAs using a transfection reagent according to the manufacturer's instructions. After 8 hours of transfection, remove the transfection media and add 8 ml of fresh media. Collect the media containing